I'm Ted Carnevale. I hope you all hear me and can see my slides.
Yes, we can.
I'm going to be talking briefly about NEURON and then work through a short example of how to use NEURON on NSG. And without further ado, let me launch right into the first slide. Many of you know about the NEURON simulation environment, but for those who don't, this is a software package which is used widely for modeling circuits and neurons models that are closely linked to experimentation, that involve biological complexities of anatomy and biophysics, and perhaps electrical and chemical signaling as well. And it is a project which is under open source development, directed by Michael Hines at Yale. Has a GitHub page entry, actually several repositories at GitHub that are related to the project, and supported by NIH and the European Brain Project. Here are some convenient URLs for you. If you're interested, you can find out more about it at these locations. We provide courses at scientific meetings, as well as independent courses and workshops and webinars. And we're in the middle of a webinar now about NEURON. The NEURON user community, as of this month, consists of more than 2,000 subscribers to the NEURON forum and mailing list. There have been more than 2,300 publications that we know of that have reported work that involved using NEURON. And in terms of source code for NEURON models, you'll find over 740 of them at ModelDB, which is a searchable database of published models. ModelDB contains also over 800 other published models as well. Well, about NEURON itself, you build models either through a programmatic process using HOK or Python programming language, or a combination of them, or with NEURON's GUI, and you can combine all of these things if you like to take advantage of the strengths of all of them. It has tools also for analyzing models to verify that what's in the computer is close match to your conceptual model. And very importantly, since these are procedural programming languages, at least with Python, you can do introspection into NEURON models, Pythonic introspection. We'll skip over the rest of this because you'd expect there to be some way of launching simulations and controlling simulation flow. You need to have ways to stimulate your models. You need to make user interfaces. The NEURON has a programmatic way of adding new functionality to its computational engine in the form of new ion channel specifications or synaptic mechanisms or means of chemical signaling and other things, including artificial spiking cells. It also has a rather powerful reaction diffusion module in Python, which can be used to deal with diffusion transport, accumulation of chemical signals, reactions among them. And most appropriate and pertinent to our present discussion, it has 3 different ways of parallelizing simulations, and we'll talk about those shortly. And these are nice links you can go to if you want to learn more about some of these things. So let's talk about what, again, it is that NEURON is useful for modeling. And the reason it's of interest is because the moment-to-moment processing of information in the nervous system involves propagation and interaction of electrical and chemical signals that are distributed in space and time. And oftentimes the equations that describe these processes are very complicated, so it's difficult to have a clear understanding of them or a good intuition about what's going on. And so when you reach that circumstance, that's when you need empirically based modeling to help you with your understand your conceptual models better. So to think a bit more clearly about this process of how it is that we develop understanding about the physical world. The physical world, of course, is extremely complex, and in some ways, in many ways, not yet completely understood. And all of our understanding about the physical world is obtained by a process of simplification and abstraction. And in any given circumstance, you're never thinking about any particular thing in the real world in all of its ramifications. You're always dealing with a subset of its properties, a subset of its behaviors, of its attributes, okay? And we're quite accustomed to doing this on a daily basis. So for example, if you're crossing the street and you see a large truck moving at you, you do not have to be a trauma surgeon or a physicist to understand that you better get out of the way or something bad is going to happen. You don't need to have great complexity in your conceptual models that you need for everyday situations. But in the course of scientific research, as we learn more and more about brain cells, brain circuits, the signals that they use, and how these things are regulated and interact, hypotheses that people are formulating about how it is that brain function emerges from these properties are becoming much more complex, to the point where, again, in order to plan experiments, in order to understand and interpret the results of experiments, it can be quite difficult. And those are the circumstances under which you need or might benefit from resorting to a computational model, okay? So if you are in that circumstance, you need to be able to make a computational model that's a very close match to your conceptual model, okay? And the utility of a simulation tool is directly related to how easy it is for you to establish and verify that match. So if you have a conceptual model that's very complex and you decide you're going to implement it in C++, you don't get much help from C++. If it's a model that's in a particular scientific domain and you use a domain-specific simulation environment like NEURON for neuroscience models, that's where you get benefit from the simulation environment, okay? All right, so an example, practical example. Here's a side view of a CA1 pyramidal neuron, and we can see lots of dendrites branched all over the place, a lot of anatomical complexity, and this cell also has a great deal of biophysical complexity, many different ion channels distributed over its surface, many different chemical signals going on inside it also. But if your experiment involves nothing more than, say, applying a patch clamp electrode to the cell body and injecting brief current pulses that are low in amplitude, so they're below the spike threshold, and then observing the resulting voltage transients, then it may be sufficient to have, as your conceptual model, this very simplified ball and stick. That is a lot of abstraction and simplification. But with such a model, it is possible to very closely replicate many experiments in which an electrode is attached to the cell body and current is injected and the resulting voltage changes are observed. Okay, what about if we really want to dig into this conceptual model? Well, it does have an analytical solution sometimes, depending on the properties that you assume for the membrane and depending on whether or not the dendrite expands or gets smaller in diameter from the cell body as you move away from the cell body and what that relationship is. But even that analytical solution itself is quite complex, and it involves functions that many of us are no longer familiar with, like hyperbolic cosine, hyperbolic tangent. And for those circumstances, then it's useful to be able to resort to a computational model. And this little block of code in Python and this little block of code in Hoke, either of these, if executed in Neuron, would recreate this model, and you could then add additional attributes as you like to be able to have as close a match as you wished between what's happening in the computer and what's happening in what would happen as a consequence of your conceptual model. All right. Now, what kind of models can Neuron handle? Well, it can handle models that have a wide range of anatomical complexity, ranging from single compartments or even artificial spiking cells to highly detailed reconstructions of biological neurons, and believe me, these can have enormous complexity, to large-scale networks whose elements may involve any combination of artificial spiking cells and simplified or detailed neuron models. In terms of biophysical complexity, these models can involve anything that is described by kinetic schemes, differential equations, or events, including commonly observed things such as voltage and ligand-gated currents, synaptic transmission, important properties of biological cells and circuits, and also it can include representations of artificial spiking cells. Now let's get back to how we represent the properties of models of biological neurons. In neurons, there are basically two kinds of signals that we're interested in. One is electrical, one is chemical. In both cases, there's something that moves, and there's a driving force that makes it move, and there's a conservation law that allows us to write an equation that describes the relationship between what's moving and what's making it move. And the forms of these equations are identical for electrical and chemical signals. They're both described by exactly the same mathematical formalism. So we're going to stick with electrical signals for right now. And here's a cartoon of a small region of a cell, small enough that the transmembrane current, that is the current leaving the cell from the inside to the outside, is uniform over its surface. And then we have these adjacent regions of the cell from which current may enter our small region by flowing through these boundaries. And all these arrows indicate the positive directions for the transmembrane current and the axial currents. And if we were to write the conservation law for this small part of the cell, we would say that the transmembrane current, which is the term on the left-hand side, the sum of the transmembrane capacitive and ionic terms, the net transmembrane current is equal to the sum of all the axial currents arriving from adjacent regions, okay? Well, what if we have a very compact cell? So it can be represented by a single compartment. So something like, say, a very small stellate neuron, whose membrane current is going to be nearly uniform over the whole surface. And say we're able to maybe attach a patch electrode to the cell that gives us access to the center of it, and we can inject currents through there and record the intracellular potential. Well, in that case, our entire model consists of a single equation, okay, where this is our transmembrane capacitive, and this is the sum of transmembrane ionic currents, and what's on the right-hand side is the injected current. There's no axial currents from adjacent regions because there are no adjacent regions. Fine, that's good and simple. But what if there is a cell with significant electrical extent? Then we have many regions. We have to chop the cell into many regions in order to make these regions small enough so that membrane potential is membrane current is uniform. And we have a different conservation equation for each of those compartments, okay? So that's an awful lot of compartments. That's a lot of subscripts. When biologists look at neurons, they don't see compartments. They see branches of cells, and they're not interested in knowing that today, when we're thinking about, say, electrical signals, when we can chop the cell into relatively coarse pieces, they don't want to keep track of the fact that, say, compartment number 6 is the cell body, because tomorrow they may be interested in chemical signals, which have need a much finer spatial resolution. And so they may have to chop the cell into many, many more compartments. So the cell body is now compartments 27, 28, and 29. And that would drive them nuts. So instead, NEURON uses the notion of sections, which are continuous length of unbranched cable. And so when you look at a cell and you see all these branches, then in your mind you do an explosion of the cell into its parts. And each of these little parts would be a section in a neuron model. And each of these sections is described by the cable equation. And of course, we chop each section into as many pieces as we need to get good spatial resolution of electrical and or chemical signaling in it. And when we've done that, just from the standpoint of electrical signaling, we end up with something that has an equivalent circuit that looks like this. So where a section will be chopped into many different pieces, each piece is called a segment. And within each segment is the node that is in the middle of the segment is the point at which differential equations are being solved that govern the membrane potential of that segment. And this electrical circuit represents roughly the equivalent circuit of this section, where these resistors represent the axial resistivity of the cytoplasm to current flowing down the inside of the section along the length of the section. This is the membrane capacity. These are the membrane capacitances of each section, and the little boxes here contain all of the ion channel representations for the section. So this thing is going to have this is going to be represented by as many ordinary differential equations as you have segments, and also as many additional ordinary differential equations that you need to describe each of the ion channels. So a given cell could be represented by thousands and thousands of ordinary differential equations, okay? Of course, what we're dealing with here, stepping back from it, is we're dealing actually with a continuous system in space that we're discretizing in space to get rid of this partial differential term on the left. This is the cable equation. This second order partial derivative is the thing that we get rid of by spatial discretization. But we're still left with the differential equations that describe the time course of membrane potential and each of these gating variables for each of these ionic currents, and maybe more ionic currents if there's more ion channels present. Okay. So fortunately, NEURON's notation is much more compact than this, so you can simply use in a few statements of Python, you can create an axon model that has the Hodgkin-Huxley mechanism in it, and that takes care of the entire model specification. When we say axon is a section, we get the basic part of the cable equation, and when we say what ion channels are present, we get all the other terms in the Hodgkin-Huxley equation, okay? So in more complicated models of more branched cells that have many different ion channels, we just end up with more and more terms on the right-hand side of this, and we have many more compartments in our model. Ultimately, we have something that we can then run simulations with. In this case, we built a user interface to this Hodgkin-Huxley model axon that allows us to see membrane potential as a function of distance along its length as the spike propagates from the left to the right-hand side, as well as the time course of the membrane potential in this model at a particular point. Okay, so again, how we build these models, we can do this with programmatic specification in Hoke or Python or through use of graphical tools. The graphical tools that are available are very powerful and very flexible. The import 3D tool allows you to import detailed morphometric data, say from Neurolucida, Eutectic, or the other SWC specification, which is commonly used. The cell builder allows you to create stylized models, ball and stick-like things, or branched, artificially branched model cells, or to deal with managing detailed morphometric reconstructions. You can add other ion channels to this detailed model cell and create cell classes for use in network models. The channel builder allows you to create model specifications of ion channels using either Hodgkin-Huxley style ODE specification or Markov model transitions and with rates that are voltage or ligand dependent. And there's another tool, for example, the model view tool, which is very useful for analyzing models and presents you with a browsable outline that shows, with a few mouse clicks, reveals very quickly what is in the model and where things are located. For example, where ion channels are distributed and how they may vary with anatomical position in the cell. Very helpful, since again, you don't want to look at model source code and try to pretend that you're a CPU parsing that when you're trying to understand somebody's procedural code. Okay, so here we have on the left Hoke statements, and on the right Python statements that would recreate that ball and stick model that we were looking at earlier. Either of these programs fed to NEURON would generate exact computational model that matches closely your conceptual model, that ball and stick. Now, how is it that either Hoke or Python can be used? Well, because really at the core of NEURON is a computational engine that has data structures and compiled routines that are specialized for dealing with the kinds of problems that arise in modeling cells and networks of cells. And the Hoke interpreter and Python interpreter allow you to write statements that are compact and human readable that access these data structures and their parameters and variables to configure whatever it is you need to simulate. And also anything you can do in Hoke, you can do with Python and vice versa. And if Python needs to access some particular Hoke syntax, it can do that. And if you're operating in Hoke and you need to access some particular Python feature, you can do that. Of course, the availability of Python means you can also bring in any of the literally thousands and thousands of Python modules that are available around the world. Whether you want to do that or if it's safe, you have to decide. And if you need to add additional data structures or compiled code to NEURON's main computational engine, you can do that with a programming language called NModel. And I'm not going to spend any time talking about NModel, but NModel is capable of specifying very detailed things in a very humanly readable way. For example, this representation of calcium-triggered transmitter release, which involves many states and transitions between them, including buffering of calcium, radial diffusion of calcium, binding of calcium to vesicles, release of acetylcholine into extracellular space, and breakdown of that acetylcholine afterwards. And it all executes at machine language speeds because it is translated to C and compiled into a linked library that NEURON reads when NEURON starts up. Okay. NEURON has several different integration methods that it uses. Its default is a backward Euler, fully implicit Euler method, which is extremely stable. First order precision, of course. But then when you realize that the parameters that we know about biological systems are really known only to a rather limited degree of precision, and furthermore, there's tremendous variation from nerve cell to nerve cell of the same kind, even within the same individual, let alone variation between individuals of a given species. First order is probably adequate for most cases. Any phenomena, any biological phenomena that rely critically on the value of some parameter probably are too fragile to be robust for that organism. You certainly don't want vital biological functions to depend on critical parameter values. Think how much variation there is in your mental acuity throughout the day or after a long night of being up and being fatigued, for example. NEURON also has adaptive integration that is very easy to use. I'm going to skip over that because if you want to know about that, you can learn about that elsewhere. NEURON's utility for network modeling is particularly strong, largely because of its ability to deal with spike-triggered synaptic transmission. One of the simplifications, one of the conceptual simplifications that people make in implementing network models is to assume that transmitter release happens when the spike reaches, propagating from the cell's spike trigger zone, propagates down the axon and reaches the presynaptic terminal, at which point magic happens and transmitter is released that diffuses across the synaptic cleft to interact with receptors on the postsynaptic cell. Now, there's no magic actually, it's just the spread of chemical signals. But this can be represented extremely efficiently. Instead of having to solve the cable equation that propagates the spike all the way from the spike trigger zone all the way down to the synaptic terminal, instead it is possible to just put a spike detector at the spike trigger zone. And when a spike has occurred, then generate an event that is going to be delivered to the synaptic mechanism after a delay that is equal to the sum of conduction latency and synaptic latency, which is about 1 millisecond, okay? And that allows extremely efficient simulation of networks because we throw away all the equations that are needed, say, we don't have to solve the cable equation at all those little compartments between the presynaptic terminal and the cell body. We also get a lot of efficiency because suppose there's a presynaptic cell that projects to many synaptic targets. If you attach many of these things called netcons, which implement the spike detector with the delay and the event delivery, each of these netcons will share a common spike detector. You don't have to monitor that same point many times to see if there's a spike there. There will be only one spike detector, and each of the netcons can have its own delay and its own weight, which is the strength of the interaction, that is how much it perturbs the synaptic mechanism, okay? Also, this strategy that's used in NEURON gives us very efficient convergence. For example, suppose we have a lot of synapses of a particular type attached to a particular region of the cell, for example, basket cell synapses attached to the cell body. There can be 50 or more of those attached to a cell body. Each of them is GABA-A-ergic, and each of them, when it opens a channel, is going to be opening a channel that has a very similar time course, is going to have an identical reversal potential, the reversal potential of chloride, of course, to any of the others. So instead of representing those 50 different GABAergic inputs to the cell body with 50 different differential equations, simply have one differential equation which replicates the dynamics of a single synapse and drive it with events converging from many different presynaptic spike sources, each with its own weight, and linear summation of synaptic conductances give you the temporal summation of synaptic conductance that you would expect. Okay, so this is extremely efficient because instead of having to deal with 50 different ODEs, just one ODE perturbed by different afferent spike streams. So to see how this works in practice, let's imagine a synapse with a fast rise in monoexponential decay. This is the key to it right down here, is this block which says when an event is delivered, what happens is synaptic conductance is perturbed by an amount equal to the weight of that connection, after which the synaptic conductance continues its monoexponential decay, which is specified by this derivative block. Okay, much more complex mechanisms are implemented and have been implemented for event-driven synapses, and they work extremely well. And in fact, this ability to deal with events has allowed NEURON models to also take advantage of event driven artificial spiking cells, such as this integrate and fire artificial spiking cell, in which every time an event arrives, the excitation variable, the membrane state variable, M, is perturbed by an amount equal to the weight of that synapse, so that if you have two afferent streams, one with a weak weight, one with a strong weight, arrival of an event on the weak stream makes the membrane state jump by a little bit. Arrival of an event on the strong stream makes it jump by a lot. And if it jumps over 1, this artificial spiking cell generates an output event and is reset its membrane state to 0, where it stays until a new event arrives. This cell takes almost no computational effort because it doesn't actually involve numerical integration. It only needs to be updated every time a new event is delivered. And after that event is delivered, you don't have to do anything more to it because you know what its future time course is from the analytical solution of the first order differential equation that describes the time course of M. So cells with our extremely simple descriptions, whose future behavior is knowable based on an initial condition problem that has an analytical solution, can all be handled as artificial spiking cells for event-driven simulation. And event-driven simulation cells like that can be mixed in networks with biophysical model cells, and they can generate events that are delivered to each other. So you can have quite elaborate constructions of network models. As I said earlier, NEURON allows 3 different kinds of parallel simulation. One is bulletin board style, where tasks are posted to a bulletin board, and the workers pull these tasks off the bulletin board, grind on them for a while. Each host that does this grinds on it for a while, sends its response back to the bulletin board, and the master then pulls these responses back from the bulletin board. Requires very little effort to program that. This kind of embarrassingly parallel simulation is very common for optimization and parameter space exploration. NEURON also can do multi-threaded parallel simulations. This doesn't really require any programming effort usually. And more pertinent to our discussion is its ability to deal with network models that are distributed over multiple processors, in which the spike source and target may actually be on different processors. Furthermore, these models may involve very large, complicated model cells that may be split into pieces and distributed over multiple processors to get load balance. And it is possible to use these features to be able to simulate quite large networks. And I will skip over how this works, and if we have time, we can come back to that. But the point is that there have been many published models that have used this feature, and benchmarks of performance have been performed with these various models, and generally they scale quite well. So that if you increase the number of processors for such a distributed model, you get an appropriate reduction in runtime that you would expect if you had strong scaling, until you get to the point where each processor has so small a piece of the original model that it doesn't have much to do. If it runs out of stuff to do between in the course of a simulation, between when it's calculating and when it's exchanging data with other processors, then you have some wasted time and you need to maybe do a little more sophisticated load balancing, which is possible with NEURON. It is assumed that before you start doing this stuff on a resource like Neuroscience Gateway, that you will have worked through those parallelization issues, such as load balance, model setup time on your own hardware on a toy or downscaled model. And it's very possible to implement models that you can, by changing a single parameter, change the scaling of. And so you can do all that debugging locally, and then when you need to use it on the production simulations, you can move your code to something like Neuroscience Gateway and run the code there. Okay. At this point, I imagine there's a gazillion questions. Maybe, maybe not. Let me see here.
No, Ted, there is nothing I think you need to answer right away.
I think you're right, Amit. Very good. So let me then, let me go to Neuron on the Neuroscience Gateway. I'm going to share that. Okay, do you see this now? It's on the Neuroscience Gateway portal, Neuron on the Neuroscience Gateway portal. You see that
Yes.
slide? I hope.
Yes, we see that.
Okay, good. All right. So fortunately, it exists now. 9 years ago, it didn't exist. And I'll tell you, we were having a difficult time with it because Neuron's ability to deal with distributed models already had existed for about 6 or 7 years. And the problem was that very few neuroscientists had access to parallel hardware that was more than, say, whatever was on your desktop, or if you were fortunate and you had a geek who could put together a small Beowulf cluster or something for you. So what we were doing oftentimes was helping people who had begged, borrowed, or stolen time from their local engineering departments or physics departments, where they might have had a few cycles left over after they finished simulating the universe. And you buy those people enough beer and maybe they'll give you an account and maybe you can play with your silly neural simulation tools. But helping people get set up on all these different platforms with all the different local policies that they used with regard to file management, queue management, et cetera, was a big pain. So it was a real relief to us when the Neuroscience Gateway was originated, because that meant that people could run their code on Neuroscience Gateway, where our programs had been installed and properly configured optimally, and where they had a nice uniform interface that Suba showed you. So who's actually been doing what? Well, we know about the 31 publications in refereed journals that we've been able to gather from users of Neuroscience Gateway. Again, we have to ask people, please tell us what you're doing when you publish. But we know there's also many, many posters and presentations at meetings that we've seen ourselves, and fortunately people are reporting those to us. And when we look at the topics that people are using the Neuroscience Gateway with Neuron to study, it includes things that range from optimization of large models of cells and brain circuits, studying mechanisms of generation of phenomena such as local field potential brainwaves and EEG, to study how it is that normal and abnormal brain function emerges from the anatomical and biophysical properties of cells, such as olfaction, learning, and memory, and epilepsy. To develop and deploy research technology, such as new methods for spike sorting, very important for people who are doing large-scale recordings from cells in behaving animals. Development of simulation software such as NetPine, which actually uses Neuron as its computational engine. Development of environments for model optimization and collaborative research. And also people are using this Neuroscience Gateway for simulations involved in development of therapeutic technologies such as stimulation of peripheral nerve or central nervous system, for, say, for example, deep brain stimulation, motor prostheses, sensory prostheses, neuromodulation. And there's a large amount of application in education, especially neuroscience education, bioengineering, and computer science. But one thing in common for all of these is before you start to use the Neuroscience Gateway, it's a really good idea to make sure that your code is debugged and you run some preliminary benchmarks so you have an idea of how much time you need, how many processors you're going to use, because it's a long turnaround. So if you have a problem that crops up when you're on the neuroscience gateway, you've thrown away time that you were allocated, and it's not a very tight debugging loop, because there is a turnaround time, much faster on your local hardware. Okay, so today for my example, I'm going to show you a NEURON implemented simulation of a cortical column involved in actually thalamocortical network. And this original paper was published by Roger Traub and his collaborators back in 2005. And they implemented their code. Actually, Roger implemented their code in Fortran on a very particular architecture machine. But it's a useful model, so it's been translated into to NEURON, not just in the original Fortran that it was. This involves many different types of model cells. The toy implementation of this that we're going to look at today has 356 cells, and the full model is 3,560. And as I said, the original implementation was very lab specific. It was designed to run on exactly 14 processes on a computer that only Roger had access to out of the whole planet. Tom Morse and Michael Hines worked with Roger to port that to G77 Fortran, and then to implement, Tom and Michael worked to implement a NEURON model with that, which would work with any number of processes on any machine that has NEURON installed. Okay, now the translation is not a perfect translation because of the way that Roger implemented things such as branching, and he did many idiosyncratic things which don't translate well to anybody else's simulation tool, but you can make sort of a first order approximation to it. And if you want to know the details of that, definitely look at this ModelDB entry and read the README files. I downloaded the NEURON implementation, unzipped it, compiled the mod files, and checked the runtimes on my own desktop machine just to make sure that I wasn't going to be unleashing the Sorcerer's Apprentice. Turned out that runtimes were quite reasonable, setup and runtimes were quite reasonable, so I didn't have any qualms about doing this with Neuroscience Gateway. So this is what I'm going to try to show you a bit about today. Suba pretty much took you through the steps, and we're going to basically just recapitulate those. I created a folder, I uploaded data to that folder. I think I might skip that because I don't want to stress my poor quality internet connection. And then configure a task, run it, check status, and download and examine results. So let me stop this particular share and start a new share. Okay. Good. Let me make sure that of what we're looking at here. You should be looking into my account at the Neuroscience Gateway. Do you see that? It says Traub 2005 up here in the upper left.
Yes, Ted.
Everybody sees that?
Yeah, Ted, we
Very
see
good.
That.
Okay, good. So what I've already done is I've already created a folder. And as Suba pointed out earlier, when you do that, whoops, I'm bounced. I'm going to have to log in. Log in. Okay, good. I've already created a folder. And as Suba pointed out, you're going to have within that a subdirectory for data. And I have uploaded the zip file for Roger's model. I got that zip file straight from ModelDB. I'm not going to bore you with showing you that. I just expanded my Firefox window. Do you see the expansion now?
Yes, that we saw the expansion.
Did you see the whole Firefox window? Good.
Yes.
Good. All right. So you can see here already I have this data object which I uploaded. And when I first went to tasks, there were no tasks. So my first thing I did was to create a task. And I'm going to create a new task. And I'm going to call this Traub trial run. Okay. And my input data will be from my data folder of Traub 2005. And I'm going to click right there in the checkbox. I hope your screens have all updated now. And you see the checkbox is selected. And I will now click on Select Data. And it says one input set. Very good. So that's the source code that we're going to use. And it was a zip file. And I know exactly the information that I need from that zip file in order to be able to specify how the simulation will run. What tool am I going to use? I'm going to use Neuron 7.7 on Comet. Okay. And if I want to learn more about it, I could click on this eye, and that would pop up a window that would tell me more about this implementation. But I'm just going to click on Neuron 7.7 on Comet. And now it tells me, it reflects my selection. That's good. Now, what are the input parameters? The input parameters are what I would call my simulation and parallelization parameters. What are they? Well, as Suba reviewed, how long do you want this to run? Well, I'm going to leave that at a half an hour. It won't take anywhere near a half an hour. I'm just going to leave it at half an hour. That's safe. The main input file name is, in fact, the file name that has to be executed by the simulator in order to launch the simulation. This is the correct choice for me. This default of init.hoc is the correct choice. If I needed to type something else in, I could do that. Do I need to run Neuron with a single extracellular layer? No, I don't need to do that. I'm not using extracellular, and I don't need to use single extracellular layer. It is in HOC. I don't have to say it's in Python. There's no subdirectories I need to specify. The command line option, I need to tell my program, my program init.hoc is looking for a command line option that tells us when to stop. And so that is a command. So it's -C is the switch. And then I do single quote, my T stop equals 100. And there's my closing single quote. And then I hit enter. Then I come down here and let me go back. Did that get set? Yes, good. Okay. So I see that my runtime that I specified is indeed 100. Very good. Fine. How many nodes? I know there's, what, 32 nodes on, excuse me, no, I know there's a bunch of nodes. I don't need any, I don't need more than one right now because I'm only going to be using a handful of cores. And I'll just let them all run on one node because I think that'll be adequate. Okay. In this case, if I wanted to, I could say, use just one core and just see how slow the simulation could be. But I'll leave it at 8. A default of 8 is fine. I just want to see that I get a result. Okay. And the name for the compressed output file, I'm going to call this test run. So there will be a zip file test run result. Okay. That's a long name, highly mnemonic. If I was doing something very systematic, I might instead include a hash code in that name just to keep track of all the parameters that I set properly and make it unique. But that's adequate for now. I don't need any advanced parameters. I will click on the Save Parameters button. Save Parameters. Now let me just look and verify that the parameters are what I want. Click on the 8 parameter set, because even though this takes a little bit of time to check these, it's going to take a long time to run, a much longer time to run the simulation by comparison to the time I used to set this up. And if I've made a mistake, I'd better catch it now. Okay, so let me go back. This is fine. Let me just click on tasks. And where is, whoa, I didn't save the task. Oh dear. Well, there's a mistake. But wait a minute, continue editing current tasks. Will that save me? Trial run. I didn't mess up too badly, did I? So let me just save the task before I destroy it. There we go. Trial run. Okay, let's just look at those parameters one more time. I'm just checking it, and I'm not going to show you it worked. I didn't destroy anything. Okay, so there I am. There it is. So this, if I want to run this, I click on Run Task, and it gives me the warning here, etc. Okay, fine. I will be patient. Click on OK. And now we're off to the races. Okay. It says View Status. I can click on View Status. And I see these task messages, and it says it's in input staging. That's very nice. I'm being told that my internet connection is unstable. So before, you probably got some garbled voice there because my internet connection does appear to be unstable. This will eventually return. Let me go back to the task list, and I'll show you one of the results. Okay. So here's one that I ran that ran on two machines, so that I could actually get a total of 32 processors and not cut into the memory that they were sharing. And here is my output file. And if I want, I could examine that in detail. And let me see here. Can I view this? Ta-da! How do I view this? Can I look at this? Click on an output file below to review its contents. Well, let's see. View current task. I don't know if that's going to show me anything. That's not the contents. But anyway, what I would do is I just download the darn thing to my own PC, expand it, and get results. And if I want to see those, let me see. Let me just stop sharing, and I can show you now on my own machine the results. Okay, share, and this is on my own machine, CD results, CD N2C32, and here is the major result, Traub, out 32 dat. Okay, and this shows me all the different times at which cells fired and the identity of the cell that fired. Okay, and I can dig into these and analyze and plot results and see if I got something that's useful to me. So that's about all I have to say. Are there any questions? Okay, if there's no questions, it looks like I'm ready to pass the baton. And at this point, I'm actually going to have to leave the webinar because something has come up in the interim, and I'm just going to have to quit now. I will be available by email afterwards, and I imagine you will see many useful things in the remainder of the show. Okay, thank you very much for your time.